Okay, for this part of the chapter, let us examine what actually is going on with the proteins that are embedded in the membrane. Now, the membrane itself is basically just a barrier. It doesn't do any work. Okay? For work to be done, for interactions to be done, there has to be proteins. Proteins are the transporters. They move things from side A to side B across the membrane. They're the receptors. They're the enzymes that do the work, that make things, break things, or the things that hold the cells tight together to form a tissue. So, we find that the proteins that are associating with the plasma membrane come in two forms. First, what are known as integral. Integral proteins are like this, what we see here. They're embedded in, they pass through the membrane. You also have, as we see right here, something that just sets either in one half of the membrane or just outside, just to the outer edge of the membrane in the cytoplasm. These are the peripheral proteins. They're at the periphery of the plasma membrane. What's interesting about the integral proteins, we see that they do have some commonality across whatever they are, whatever they're doing. They have a certain aspect to them that we see right here. One there, three here, quite a few there. This is going to be a region of amino acids that are extremely hydrophobic. Now think about it. If you're a hydrophobic molecule that's part of a bigger polypeptide or a bigger protein, where's the best place to hide in a cell? With other hydrophobic compounds, other hydrophobic molecules. And what's the most hydrophobic thing there is? The fatty acid chains. That's right. The fatty acid chains are the phospholipids. So we're going to see that amino acids, such as we see here, their R groups are extremely hydrophobic, do not want to play with. So they're going to embed in the plasma membrane. We're also going to see carbohydrates come into play. Carbohydrates that are going to be either added, as we see right here, added to a phospholipid, or, as they're showing right here, added to an integral protein. Glycolipids, if it's added to a phospholipid, glycoprotein, remember glyco means sugar, glycoprotein, if it's added to an integral protein. We're going to find that this occurs in the Golgi. Remember, it's the Golgi that does the final tweaks, adding of sugars and processing. This is what it does. Purpose of these glycoproteins, glycolipids, well, cell recognition. Make sure the cells recognize each other. Make sure the cells know what's there, what isn't there. Now, take a look at this. This is a little bit of immunology for you. The glycolipids, the glycoproteins, they act as receptors. They act as antenna. Here you see a glycoprotein, a glycoprotein known as CD4. CD4 sticks out of the cell. Sticks out of the cell so that its appropriate signal, whatever that's going to be, as it's floating by, will attach. It will then cause a series of reactions in the cell. Cell starts to do something, cell stops doing something. That's the purpose of a receptor. Now, over the millions of years of evolution, as we mammals have been evolving, so have the viruses. Viruses such as shown here, HIV. HIV has evolved to hijack CD4. It hijacks CD4, triggers its own uptake, so it can now infect a CD8 cell.